Okay, so let's start. So last week we completed chapter 1, and today we are going to start chapter 2. So chapter 1, actually we talked about the invention of uh, microscope, right? That how we could, find out the diversified microbes, community of microbes. So, okay, somebody is joining. Okay, so now at this moment, we have microscope on our hand, okay, and we have an idea about the microbes, their diversity, and we have a classification system, so we can classify them, we can have nomenclature system, so we can give them name, okay. uh, but the problem arises that when we really want to go to deeper into uh, uh, the microbes, um, means like when we really want to see the very minute DNA of chromosome. Okay, basically, uh, when we really want to work detail on the structure of the microbes, uh, um, what happened? Give me a second. Somebody is joining. So basically, when we really want to study the detailed structure of microbes, um, then we realized that mm, only microscope, having microscope is not enough. Okay. Um, we actually, from that time onwards, we started working with the light. Okay, we realized, or we discovered that um, all this microscope, at least that time, during that time, we had on our hand, uh, all were depend on the light. So this much we realized that lots of things actually depend on the characteristics of light. Okay, and we also found out that if we know the properties of light, okay, if we know more how to use light, okay, or how differently we can use light, then we might improve the image quality, okay, and we might can see the more detailed structure of the microbes, okay, or more detailed characteristic features of the microbe. And from that time onwards, we started working on the light, okay. So this chapter, it's how we see the invisible world, okay. Of course, we can see the invisible world with microscope, but now we are in advanced stage when along with the microscope we started playing with the lights, and we discovered so many different concepts how to utilize light differently. Okay, so this chapter basically talks about different types of microscope and how we utilized light inside all those microscope. um, there are few microscope where we really do not use light at all, okay. um, there are microscope where we utilize the fluorescence characteristics. So all together, this chapter basically will talks about different ways to observe the microbial world. Okay, and we are going to talk about so many different types of microbes, microscope, different ways how we actually fix one specimen, or we prepare specimen for a specific microscope. So altogether, it's entirely about the different types of microscope. So, okay. And you can see my screen, right? Do you see my screen?
Yes, ma'am.
Yes, ma'am.
So in this slide,
Yes, ma'am. Sorry.
thank you. Okay, in this slide, you can see both are two different pictures, but two different types of image. The first one, it's called bright field. The image actually came from bright field microscopy. Over here, you can see that the image are really darker, and the background is really bright. Okay, so in this picture, dark image with bright background. In this picture, you can see image is very bright, but the background is completely dark. Okay, in both the picture over here, it's I think it's Bacillus, one rod-shaped bacteria. Over here, you can see that Spirilla, or spiral-shaped bacteria. Okay, only difference is we captured these images from two different types of microscope. Now the the first one, it's called bright field microscopy, and this one is called dark field microscopy. Somebody is joining. So bright field microscopy and dark field microscopy. Okay, so bright field microscopy. As I told, that really dark image, but brighter background. Background is very bright. Versus over here, background is completely dark. How we did it? So if we compare these two pictures, the difference between these two, the background over here, the first one, it's really bright background. Here, background is completely dark. Okay, so if you think, if I ask you, just from general sense, or from by seeing the image, okay, and by knowing the microscope characteristics, what do you think? How we actually made this? To make a bright background, what we have to do? Anybody can tell? Just look at the background over here. The second one, it's completely dark background, but over here it's really bright.
Fluorescent.
No, it looks like fluorescent. No, but you got the concept. Over here in this picture, basically, because the background is so dark, it looks like that it's fluorescent, but it's not. It's really the contrast. Okay, the contrast is so dark, or you can say so bright actually. That's why the organisms or microbes are very clear. So the difference between these two picture, these two image, basically what happened over here in the first one, bright field microscopy, we have enough light. Okay, we supplied enough light to illuminate not only the specimen, even the background. Second one, in the dark field microscopy, what we did, we didn't supply enough light so that the background is not so lighted. Basically, there was not enough light, and that's the reason the background is completely dark over here. So in this picture, the first picture, we provided enough light both on the specimen and on the background. In the second picture, we provided light only on the specimen. Okay, we modified the microscope in such a way so light only could go through the specimen. Okay, that's the difference between these two. I made this as my first slide to show you how we actually started playing with the light to observe the microbial environment or microbial community from so many different angles. Okay, so what is light? So before we go to the detail of the microscope structure, first we really need to know the properties of the light. Okay, so what is light? Light is an electromagnetic radiation that behaves like waves. It's electromagnetic radiation, it behaves like waves. So wave always moves like this way. Okay, light is an electromagnetic radiation that behaves like wave. So when it is like that, when it is a wave, all wave must have the median. So this line is called median. The length, so this is the peak, these are all peak. Okay, the lower peak is called Strout. Now from the median line to the peak, the height is called amplitude. Now the rate of oscillation. So over here, let's say, so when basically the rate of oscillation, that's called frequency. Okay. Now rate of oscillation means how many peaks or how many waves it can make per unit time. So let's say over here we have 1, 2, 3, 4, 5 peaks per second. So how many waves one light can make per unit time? It could be unit time, means it could be second, it could be minute, it could be hour. That's the frequency. So rate of oscillation, all the frequency. People are still joining. Okay, so over here in this picture, I draw what's the frequency for this one. So this is picture 1, let's say. So what's the frequency of picture 1? It's 5 per second. Okay. Now, if I draw another picture, let's say over here. So if and so in this one, number 2, second picture, how many waves are there? Just 2. So What's the frequency over here? It's 2 per second. Okay. Now the distance between the peaks. So from here to here, this is the peak, this is the peak. So distance between two peaks are called wavelength. Okay. So if we compare these two pictures, so the frequency over here, number one picture, it's 5 per second. A frequency of second picture, it's 2 per second. Okay. Now, if we compare the wavelength, so over here, the wavelength is from here to here. From here, wavelength is from here to here. So what's the difference between these two pictures? Over here, in every second, this radiation, this ray made 5 waves. Okay, and that's the reason the wavelength is much shorter than here. Here, for this radiation, for this ray, each second it just made 2 waves. Okay, just 2 peaks. So wavelength is much higher. So our time limit, just this much distance it can cover from here to here, this much, okay, per second. So every second that light can travel from here to here. Now in this case, the wavelengths means the distance between the peaks. So wavelengths is much higher. It's really large wavelength. If the wavelength is high, then the number of peaks goes down. Okay, number of peaks goes down means the frequency goes down. But if we keep the distance same and we reduce the wavelength, then frequency increases. Now, this is the wavelength from here to here. But over here, frequency is much higher than this picture. Okay. Now, if we represent frequency by f, lowercase f, and wavelength by w, then we can draw a relationship that f varies us with 1 by w. Means if frequency increases, wavelength decreases. On the other way, if wavelength increases, frequency decreases. Okay, so that's the relationship between the frequency and the wavelength. Okay, so did you get it? The relationship between frequency and wavelength?
Yes, ma'am.
All of you, do you have any question?
No, ma'am. Thank you.
So you got frequency, right? Frequency means rate of oscillation, means how many peaks it is going to make. That's the frequency. And wavelength means the distance between two peaks. So if you compare these two pictures, so in both the cases, the distance covered by the light is same. Both are same, the distance. But over here, frequency is less, means it just made 2 peaks. But over here, frequency is really high. It made 1, 2, 3, 4, 5 peaks. This part you got, right?
Yes.
Okay, so now over here, so frequency is less means the distance between the two waves or between the two peaks is longer than this distance. Okay, so then can we say that if frequency increases, high frequency means shorter wavelengths, low frequency means longer wavelengths, larger wavelengths. Okay, so frequency, if we denote frequency with F and wavelengths with W, means we can represent the relationship between frequency and wavelength like this way. So frequency, if frequency increases, wavelength decreases. If frequency decreases, wavelength increases. Okay, so F means that's the frequency, W means that's the wavelength. Clear?
What is the word after the 2-sec, and then 5-sec? What is that word?
That means in this picture, it just this light ray just made two peaks. So per each second, within 1 second, in this picture, this light rays made two waves. Okay, just two oscillation per second versus
Oh, okay.
5 oscillation per second.
Thank you so much.
Okay. Now, as I told that a light actually behaves like waves. So all the characteristic features actually follow the characteristics of waves. Now, what happens when light falls, light waves falls on a surface? Then most of the time it actually bounces back. So let's say this is this is one surface. Okay, so this is the surface and light rays are falling on this surface. Few things can happen. It can bounce back, or this light can completely pass through the surface, and our median is over here. Okay, so light rays can fall on the surface and it can bounce back. That's one condition. Second, it can completely pass through the surface or the material. That's the second incident that can occur. The third one, once light fall on the surface, it can be scattered in completely different direction. So these are the three situations can happen when light falls on a surface or light tries to pass through any material. Now, these three situations can happen based on the material, what type of material it is passing through. So our first type of material, it's called opaque. The opaque material does not allow light. Light cannot go through the opaque material. So what happens in this case, when light falls on the opaque material, either that material completely absorb the light energy, okay, So let's say one opaque example of one opaque material is, let's say, book. When this is a book and light wants to pass through this book, what happened? Light cannot go through it. So the book, what it will do? It will absorb all the light energy. And as a result, what will happen? The temperature of the book will increase. We know that energy cannot be destroyed. One type of energy can be transferred into different type of energy. So over here, this is one possibility, is it can happen when light passes through the opaque material, that material completely absorb the energy. So that can happen. On the other hand, when light can move through the opaque material, sometimes what happens, this opaque material, they reflect back some light and they got scattered. Okay, so that's the opaque material. Book, table, chair, all are example of opaque. Now, second type of material, it's called transparent material. Transparent material, so example of transparent material is air, water, you can say glass. So when light passes through the air, if this is the air, what happened? Light completely goes through that material. Same thing happened with the water and the glass. So that's the transparent. So almost all the light rays can move through that material. Now, number third category, they are called, they are called translucent. In this category, what happened? Light rays cannot completely move through that material. So material actually block most of the light and it allow few of the light rays to move through. And most of the light, either it absorb or it actually help light to be scattered. Okay, so these are the three different types of material based on the density of that material and how light actually behaves when they pass through these types of materials. Now for the opaque, what happens? Opaque material, light cannot pass through it. So opaque, what it does, all the light, it falls, it actually either it bounces back or it get absorbed by the material. So when the light falls on a surface and it bounces back, then that's the characteristic features of light and it is called reflection. Now, absorbance. So as I told, the characteristic features of opaque material, that it just reflects back very little amount of light. Most of the light it actually absorbed, okay, and produces heat energy. So it was light energy and it converts into heat energy, and that's the reason the temperature of this material increases. So it absorbs, that's the characteristic features, and it is called absorbance. Okay, so occurs when a material captures the energy of a light wave. every time when absorbance occurs, means one type of energy converts into other type of energy. Like over here, I gave you an example in the case of book, the light energy converts into heat energy. So that's the absorbance. Transmittance occurs when light passes through a transparent object. So it falls on the object and it completely moves through that object. Okay, so when light travels through a material, like when it can travels through the material. So if over here 100 rays falls onto this surface, and almost 99% of the light rays can pass through that material, that's the transparent object. And it can happen in air, water, and gas. Okay, glass. Translucent objects, example, you can say the frosted glass. We can see the light, but we really cannot see any image that comes through the frosted glass. So that's the translucent, okay? Because we can see the light very little bit, because it allow only a very little amount of light to move through. Most of the light get scattered, okay? We can see the image. Now, to create an image, what happens when it is a transparent object, and light completely moves through that object, that light cannot make any image. Image is formed when light is scattered. So when light passes through the opaque object and the translucent object, then we can see the image. Okay. When it blocks the light partially or completely, it can make the image. But when light can move through that object, then that object cannot produce image. This is the very fundamental characteristic features of light, and you really need to understand this line. If you understand this line and remember this concept, then it would be very easier to understand the microscope concept, okay? So just remember, image only is produced when light get scattered from the object, okay? Or light gets scattered from any surface. Then only we can see the image. Now, another characteristic, let's say over here I can draw. So it's a let's say it's an opaque object. So light falls, and most of the light are going back, bounce back. Okay, so so 3 rays are here, 3 rays are there. Now, and you can see that all the light are going in one direction. So if I try to extend, this light will not merge together. Okay, these rays are not going to merge together. You can enlarge these rays all the way up to infinity. Now, what happened? Not all the lights are going to move in the same direction. On the opaque object, there are few portion or certain amount of light. What happened in that case? So they falls and they get scattered. So basically, this light goes in different directions. So when this thing happens, so they are going in so many different directions, okay, few of the light rays, they fall on the same direction, they merge. Sometimes it happens. So when this thing happens, like when all these are scattered light, okay, scattered, you can say scattered rays. So when light are scattered, sometimes they fall into each other's path, they merge, and they can interact with each other. And this thing only can happen when the light are scattered. And these characteristic features of light is called interference. Okay, so light waves can interact with each other, and make sure to remember this thing that they can interact. Light rays can interact with each other only if they are scattered. Otherwise, they cannot. You can see these rays. As I told, you can extend all these rays all the way up to the infinity. They will not merge. But when they are scattered means they are not following a scientific rule. They are not following the scientific wave pathway. Okay, they get distracted, and that's when they are scattered. So if light are scattered, then they can interfere with each other. And this property is called the interference of light, okay? Very interesting characteristics. Now what happened? Let's say this is one light ray. And another one over here, okay. So over here, we have two light rays, two scattered light rays. If you see the peak of this light actually match with the peak of these waves. Okay, so basically they actually fall into each other's pathway, and they are following the same path. Do you see? So over here, both the trout are together, both the peaks are together. So in this case, when these two light waves, they are on the same pathway, when they are interfering and they fall upon each other path, then the intensity is increased. Okay, in that case, it increases the intensity. And in that case, this type of interference is called constructive interference. Okay, now another type of interference where this is one light rays, okay, and another one is this one. In this case, what happened? You can see that this trout actually is going to match with the peak. Okay, you can see these are merging. So in this case, it actually decreased the intensity, light intensity. So one light ray actually minus out the other's intensity. In that case, this type of interference is called destructive interference. So basically, when light waves can interact with each other, and these things happens only when the light are scattered, and that is called interference. And there are two different types of interference. One is constructive interference and the other one is destructive interference. Basically, when two light falls onto each other's, either they merge, either they add up the intensity with each other, and that's how they actually increase their intensity, or they minus out each other's intensity. And in that case, that's the destructive interference. Okay. One more interesting characteristic features of the light is diffraction. If light falls on a very small, minute objects, they get distracted. Okay. That's called diffraction. Or you can see light can move, how light actually can move through a hole. If it is a dark room and there is a small hole on the wall, you can see the sun rays is coming, and you can see all the different frequency of light of that sun rays. Okay, that characteristic features is called diffraction, because just one ray get scattered. Okay, and all the different frequency of light, they get separated from each other, and that's how diffraction occurs. So do you have any questions up to this? Anybody? Did you get up to this?
Yes, thank you.
So should I move to the next slide? Are you clear? Property of light?
Yes, ma'am.
Okay. Now the reflection. So okay, let me go back. Reflection, I told you what happens. So light falls and it bounces back. It goes back. Okay. Means for reflection, light remains on the same media. So light is, in that case, light over here, light is coming from the air. It's falling on the almost same category of surface, and it is actually going back to the same media, which is air, or maybe water to water, glass to glass, like that. Refraction occurs when light actually moves through two different media or three different media. So over here you can say, so this is air, and this is water. So when light is coming from the air and it is entering inside the water, so it can change its speed. So this is the median. Okay, so light is moving from one media to another media. When they are changing medium, they actually change their speed. And because they are changing speed, their direction is also has to be changed. Okay, so, and that properties is called refraction. Usually what happens when their speed is higher, when speed is high, they actually moves away from the median. So this is the median. This dotted line is called the median line. Okay. So when the speed increased, what happened? They actually goes far away from the median. But if speed decreased, then they goes towards the median. Okay, and that's another characteristic features of light, and it's called refraction. So the speed of light is highest in air. Okay, so speed is highest speed you can see on air, and then it's water, and slowest in the glass. Okay. Refractive index, ratio of speed of light through the vacuum and through which it passes. So how do you know that the light will change its direction in which way? Okay, to determine that, or what would be the speed of the light, we have one mathematical formula that's called refractive index. What we do, we compare the speed of light of a media with the speed of light inside the vacuum. Vacuum means empty space where there is nothing. Okay, and we have a constant fixed rate over there, rate of light. Every time we compare that amount with the speed of light in any other media, like let's say we want to find out the refractive index of let's say the glass. So what we do, we actually divide the speed of glass by the speed of light inside the vacuum. Speed of light in glass and speed of light in the vacuum. So that's the ratio of the refractive index. So refractive index actually indicate, if we know the refractive index, then we know the directions of the light. As I told, when speed increases, the light actually goes far from the median. Basically, this angle becomes larger than this angle. Okay, so this angle is called incident angle, and this is the refractive angle. So when speed increases, usually in case of reflection, I always equals to R. So basically what happens, let's say this is the reflection and this is the medium. Every time this is I, and this is R. So these two are equal, I equal R. But when it is refraction, when they change the speed, when the speed is higher, then this angle, the refractive angle, is larger. In that case, what happens? The R becomes higher than the I. So that ratio is, it actually get changed in case of refraction. And to determine all these, we must need to know the refractive index. Electromagnetic spectrum. So basically what happened, different types of radiation, electromagnetic radiations. So each light ray, they have their specific frequency, specific wavelength. Okay, now all these different types of light rays with different frequency, different wavelengths, they together make electromagnetic spectrum. If that electromagnetic spectrum, you can see if one light ray falls, so let's say this is the prism. Okay, if one light ray falls onto the prism, you can see that it actually breaks down into 7 different rays. Why? Because all of these light rays, they have different frequency and different wavelength. But with our naked eye, we cannot see them separately. We can see them only when they can fall into the prism that actually separate them. Okay, then only we can see them. So all these different frequency of light rays and different wavelengths of light rays together, they make the electromagnetic spectrum. So basically the wavelength and the frequency varies from here to here. So many different ranges of wavelengths and frequency are there. Visible light, the light that we can see, is actually the part of the electromagnetic spectrum. That visible light also have a specific frequency and wavelength. Okay, our eye only can detect that range of frequency, and that's the reason we can see them. Let me go back over here. So you can see this is the electromagnetic spectrum. You can see that there's so many different types of frequencies and wavelengths are over here. So wavelengths, cosmic radiation. Okay, you can see the wavelengths at 10 to the power minus 8. This part. 10 to the power minus 18 is the wavelength for the cosmic radiation. So when the wavelength is 10 to the power minus 18, now at this moment, make sure you remember that if frequency increases, wavelength decreases. So wavelength is all the way towards minus side, minus 18. Okay, very, very minute wavelength. So frequency is really high, 10 to the power 24. Versus over here, just let's say over here, infrared, okay? Frequency, wavelength is 10 to the power minus 6. So over here it's minus 10 to the power minus 6. So the frequency is 10 to the power 15. Okay, so I hope you understood the relationship between the frequency and the wavelength over here. X-rays frequency is more than 10 to the power 21. Okay, very high frequency. So wavelength is very, very minute. It's 10 to the power minus 12. So that's the relationship between frequency. This is F and this is small w. So that's the relationship between frequency and wavelength. Now energy. If frequency increases, that means energy increases. Every time frequency goes with the energy. Okay, high frequency ray means it has high energy. So we have, if we go by the frequency, you can see in this slide that from 10 to the power 24 to all the way to the 1, that's the range of the wavelengths. So many different types of light rays are there. Cosmic rays, gamma rays, X-rays, ultraviolet, infrared, radar, television and radio broadcasting wave, AC circuits wave. Okay, now all together they actually make the electromagnetic spectrum. Okay, so now over here within this spectrum, the visible light falls into this area, almost at the middle. So we cannot observe this part, this side, left side. We cannot observe the right side because of the high energy or because of the low energy, high frequency. We only, our eyes, whatever the rod cell and cone cells we have inside our eye, we only can detect this much, the range of frequency from here to here, and they are the visible light. Lens actually one object that has a curved surface. Okay, and that actually, if light falls onto that curved surface, it collects all the light rays, and what it does, it actually helps all the light rays to fall onto one point. And that point is called the focal point. And the distance between the medial and the focal point is called the focal length. We have two different types of lens. In this book, we are actually for the microscope purpose, actually, we usually use two different types of lens, convex and the concave lens. Convex lens, what happens? So make sure you know that this is the front side. So we are talking about this lens, okay? So this is the front side and this is the back. Gosh. This is the back side of the lens, okay? Because you can see the light is coming from this side. So that means this is the front. So when light falls onto the convex lens, what happened? It collects all the light and it gathered all the light on the back of the lens at the focal point. And that's the characteristic features of the convex lens. Now for the concave lens, what happens? When light falls onto the concave lens, it collects all the characteristic features of lens is it actually collect, try to accumulate as much as light possible, and that's the good quality lens, means it's actually collect high amount of light. So over here, for the concave lens, light falls onto the concave lens, but they actually reflect far away from the median. Okay, so if over here we have our median, so this is our median. So in this case, concave lens, light falls and they moves towards the median, because median goes like this. So this is the median. Okay, so they move towards the median, but on the back side of the lens. In this case, in case of concave, light falls, okay, and they goes far away from the, actually they moves away from the median. Okay, in that case, what happened? If we draw an imaginary line by tracing all the movements of the light rays, then we see that all the light rays actually merge on the focal point, which falls on the front side of the lens, which falls on the front side of the lens. Okay, so what's the difference between convex and concave? In case of convex, the focal point is on the back of the lens. In case of concave lens, there is no true focal point. We have one imaginary focal point, but that actually falls on the front of the lens. Now so we are done with the physics part. Okay, this chapter is actually biophysics chapter. So we are done with the most complicated physics part. Okay, so do you have any question till now? Rest of the chapter is very easy. Do you have any question, anybody? No?
I do have a question that's just kind of in the back of my mind, but it's just a general question.
Yeah, tell me.
About how you test. I don't know if that's appropriate to ask right now. I'm trying to wait till the end of class, but that's one of the things is trying to figure out what exactly you're looking for, where we should be studying the most at.
I am looking for each and every detail.
So would that mean that anything that you go over while we're doing this PowerPoint is understandably that no? Yes? Like whatever you go, okay, sorry.
Whatever I go over, now during there, whatever I wrote on my PowerPoint, and whatever they are inside my book, if I can give questions from anywhere.
Right, so the study guides that are for each chapter, if we are going over that considerably, would we be okay with the test?
Yes, but if you notice that study guide, okay, it's a broad, kind of broad, like, okay,
Right.
the properties of light. So properties of light, whatever I said, everything, all are included under that category.
That makes sense.
Okay. So if I ask you the interference, okay, so properties of light, one of the characteristic features of light is it has interference characteristics. Okay. Now, you can just memorize the definition. Okay, what is interference? When light falls, they interfere with each other. That's the interference. But you have to understand, as I told, that there are two different types of. Oh my God, my phone. Let me. I'm sorry. Very sorry. I forgot to change it to silent mode. Okay, so so interference, okay? I told you that there are two different types of interference, constructive and destructive interference, okay? You really need to understand why it is constructive and why it is destructive. If you understand that, then I know that you will understand the different types of microscope, like phase contrast microscope, the principle of phase contrast microscope. Okay, so each and everything are interrelated, and you have to find out that how it is connected. Okay, how that interference is connected with this chapter, why we are studying about that interference, properties of light. Why inside your book it's not that elaborate? Okay, what is reflection, what is refraction, what's the difference between reflection and the refraction, and how it is connected with all three different type of objects. I went a little bit of detail. Why? So that you can understand the concept. If you just read without understanding, what will happen? It will not make any sense. And after reading the text, reading this chapter, you will feel, okay, so why should I know this definition, absorbance? So why should I know how it is related with the microscope? Reflection, how it is related with the microscope? If you can give answer of that, if you understand how these words like reflection, absorbance, transmittance, interference, all these properties of light are related with the microscope, that means you got the concept. After reading the text, if you feel that it does not make sense, that means you didn't get the concept. Did you get me?
Yes, ma'am. I understand.
So make sure when you are reading, make sure try to understand the concept. And that's the reason, I am going over the lecture. First few chapters, all are related with each other. And sometimes you will feel that it does not make any sense. So why I am going to study the definition? But all are actually interconnected, and you have to find out how they are connected. Means you have to find out that relation. Once you can find out, you are fine. Okay, so remember over here I told in this slide, I told very important point. I told you you are not going to find out that one inside your book. I told that when light are scattered, only then we can see the image. Okay, you are not going to find out this concept inside the book that easily. But if you understand all these characteristics features, it's there. Okay, my job is to make it clear to you, and that's what I'm trying to do. So whatever I'm saying, each and every line is very, very important. If you don't get the concept, email me or text me. I will tell. I'm ready to tell again and again. Okay, but make sure that you understand that. Okay. Should I move, Mike?
Sure.
So did I give you give you the question you answered? Did whatever you asked me, did I answer you?
Honestly, it feels like it's a little too open-ended because I'm trying to figure out a way to, I guess, study the direct information. Like, I get exactly what you're saying, but it still feels like right now it feels like I need to be memorizing every word of the text.
Exactly. That's what I want to tell you. Go by the detail. Each and every detail is important for
Okay.
this course. Okay. Make sure first check the PowerPoint. All the important things are included over there. And then read the text.
Okay.
As I told that you really need to study in detail. Okay, so should I start the next part, next section? It's microscope. Okay.
Yes, ma'am.
So microscope. First question comes in mind that what makes a microscope that we use in the research lab? Okay, like genetic research, virus research project. That microscope versus the microscope we use in the lab. Why the microscope that we use for the research purpose, it's much better than the microscope we use in the regular laboratory? Or what makes those microscopes better, research microscope better? So basically, when you want to buy a good quality microscope, what are you going to look for? Okay, so like when you go to buy your clothes, so okay, so I like sky blue, so I'm going to look for the sky blue. Okay, I know okay, so this is the cheap material, this is really nice material, this is soft. So all those are characteristic features, the color, the material, the softness, all those are the characteristic features of your dress, of your t-shirts. For microscope, there are two very, very important characteristic features are there. One is the magnification and the other one is the resolution. Magnification, that's the ability of the microscope to enlarge the image. Okay, how much big image that microscope can make, that's the magnification. Okay, it could be 10 times, it could be 100 times, it could be 1,000 times enlarged. Like our regular microscope that we use in the lab, it increases 1,000 times. Okay, the object, whatever the actual size of the object, and the image you see, that image is 1,000 times larger than the object. So that's our regular lab microscope. So that's one quality, that how enlarged image that microscope can make, and that's the magnification. Resolution is the distance between very close two objects. Okay, let me draw. Okay, so let's say this is the bacterial cell, and it is the DNA, and over here it has the RNA. Now, at this moment, I actually can see the two dots. One is DNA, one is RNA. But over here, if I say this is the DNA and this is the RNA, and left hand side, this side, it looks like just one object. Why? Because those two objects, both the DNA and RNA, they are so close that my eye cannot differentiate. My eye cannot find out the distance between those two objects. Okay. Now, one of the job of microscope is to separate out these two close objects from each other. Okay, and that's another characteristics of microscope, and that's called the resolving power. So resolution of a microscope or lens is the smallest distance by which 2 points can be seen and still be distinguished as separate objects. So if the microscope can separate out these two objects, that's very characteristic feature, and we call that feature as resolving power. And the distance between these two closely related objects, they are very close, but still we can separate them out that they are two different objects. That quality of microscope is called the resolving power of microscope, and the smallest distance between two objects is called the resolution. So basically, two characteristic features of microscope. One is the magnification and another one is the resolving power of the microscope. Okay. So resolving power, what microscope does, it actually separate out the smallest distance between two objects, and the smallest distance is called the resolution. Okay. I hope you got the relationship between the resolution and resolving power. So the capacity of microscope to make prominent the smallest distance is the resolving power, and the smallest distance between two closely related objects is the resolution. How we measure the resolving power? Wavelength of the light divided by 2 into numerical aperture of the lens. Okay. Numerical aperture is that's the characteristic features of the objective lens. Means it actually describes the relative efficiency of a lens in gathering the light rays. So how much light it can gather, that quality is called numerical aperture of lens. So to find out the resolving power, what we do, we multiply the numerical aperture with 2, and then we divide the wavelength of the light with that number, and that's how we find out the resolving power. Now, usually this is the lab question. For lab final, I usually give to find out the resolving power. In that case, I'm going to give you the wavelength of light. Okay. Numerical aperture, if you see the objective lens, along with the objective lens power, it's written over there on the objective lens. You can find out the numerical aperture of that lens. Okay, so it's in the microscope. Oh, so the higher the numerical aperture, the better the resolution. Now in lab, with 100x objective lens, we actually use immersion oil to increase the numerical aperture. So numerical aperture means the capacity to gather all the light rays. Okay, and immersion oil has high. It actually increases the numerical aperture of that lens, and that means we can collect a a really high amount of light. Basically, we can focus the specimen with sufficient light. Okay, so now resolution, that characteristic features of microscope actually depends on two factors. The first one is the wavelength. Shorter the wavelengths, better the resolution. Okay, that's number one. Number two is the numerical aperture. The higher the numerical aperture, better the resolution. So in this picture, you can see these are the longer waves, longer wavelength. All these light rays with longer wavelengths, they cannot actually penetrate through all the small areas of these fingers. But if the wavelengths are shorter, then they can penetrate through. Basically, they need really small area to pass through. Okay, so that's the reason shorter wavelengths can produce the detailed image. You can understand easily, from this picture. Oil immersion lens, how it actually increase the numerical aperture. And as I told, that high numerical aperture means high resolution, and high resolution means we actually can see two very closely related objects clearly. So basically, we can see the detailed structure. You can understand the bacteria, they're so minute, and inside that cell, we have to find out the DNA. We have to look for the RNA. We have to look for the rRNA, ribosomal RNA, how bacteria is making protein synthesis. So if we really want to observe all those details, then the microscope must have high resolution capacity. Okay. To increase the resolution, we use the oil immersion lens for our lab. Okay. Because immersion oil and glass have very similar refractive indices. Refractive indices means the properties of light, how much it get refracted from the median line, actually. That's the properties of light. So immersion oil and glass have very similar refractive indices. Okay, there is a minimal amount of refraction before the light reaches. Basically, let me show you. Okay, so remember when I talked about the refraction, I told that when light moves from one media to another media, it actually changes its speed. Okay, and that's the characteristic features of the light. It changes speed means it actually changes the directions. So what happened over here? So this is the air. This is so let's say this is the slide. Okay, imagine this is the slide. This area is the slide. You have specimen over here. Light is coming from here, and this is your objective lens. To make the image, as much as light rays can move through the objective lens, it will make better image. So our target should be to move almost all the light through the objective lens. This is the objective lens. Okay, so this side, it's air. We don't have any oil, immersion oil. Okay, so and the specimen is over, let's say over here. So light is going, so this is from air, then it falls into the glass. Okay, and then again it enters into the air, then it goes through the specimen, air, and then again it enters inside the glass. So many times actually it changes its direction. And when it does, when it changes its direction and the speed, we are actually going to lose a huge amount of light rays. But in this case, what we do, we actually we add immersion oil on top of the specimen. And basically you can see that all this air over here actually is covered with the immersion oil. So in this case, what happened, light is coming through the air and then it fall onto the glass, and it goes through the immersion oil, and then it enters inside the glass. Now, the refractive index means how much it will bend, how much it will scatter. That quality or that character of glass and immersion oil are almost same. So if we add immersion oil, light feels like it is going through the same media. Okay, it feels like it's the same media. So bending or scattered of light is much less here than the here. Okay, so basically if we use the immersion oil, then we can collect really high amount of light rays through the objective lens, and that was our target. Okay, so the image quality becomes much brighter and clearer here. So that's how immersion oil actually help us to increase its resolution, because we can collect more light. Now history, we already studied all those. I talked about the history of microscope in the previous chapter, so I'm not going over it. Now the parts of microscope. We have three lens system. Okay, one is the ocular or eyepiece. So over here, the ocular or the eyepiece is the lens that remains in close to your eyes. That's the first one. Second group is the objective lens. Okay, over here. And the third one is over here. You can see that lens, that's called the condenser. These are the three sets of lens we use. So three sets of lens. One is the ocular, objective, and condenser. We just have one condenser, but for microbiology, we have four objective lenses, and ocular lens, we just have two, right and left ocular. The power of the ocular is 10x. For objective lens, we have four different, and the power is 4x, 10x, 40x, and 100x for objective lens. So you can see that all these are the objective lens. All these objective lens are actually, there is a knob, this black one, that's called nose piece. All these objective lens are attached with the nose piece. So basically nose piece actually carry all these objective. If you can, it's like a rotating knob. You can move the nose piece to actually change the objective lens. This is the arm. This is the light source. Just immediately above the light source, we have the condenser, but from the below the stage, you cannot see the condenser over here. You can see the condenser from above the stage, from here. Now outside of the stage, there is another rotating knob like this, black knob over here. That's called Irish diaphragm. Okay, you can actually adjust that knob. You can see that handle like structure. So you can move that knob to increase or decrease the amount of light that actually can go through the objective lens. Okay, we have a light switch over here, and over here another knob. You can increase or decrease the amount of light. Both sides of the microscope, attached with the arm of the microscope, you can see two knobs. One is large and one is small. Large one is called the quartz focus knob, and the smaller one is called the fine focus knob. When we move the coarse focus knob, we can move the stage up or down. Okay, and the fine focus is only for fine focus. So when we want to focus the microscope, we actually work first. You work with the coarse knob to fix the distance between the objective lens and the specimen. I think, and over here attached with the stage, actually there are two knobs. By moving these two knobs, you can move the stage either front or back, or maybe right side and left side. This is the stage. On stage, you can see there are clip. This is the slide holder. Okay, so you can fix the slide by using this slide holder on the stage. Okay, so how much time do we have? 7. Oh, okay. Now the light microscope. Okay, so that we use in our lab. Most of the laboratory microscope are the light microscope. So it is light microscope because we use light to illuminate the specimen, to see the specimen or to make the image. We use light, and that's the reason it's called light microscope. Now, what happened? I am going here, back here. Okay, so if you see the, where is my pen? Okay, here. So this is the light source. Okay, our specimen is over here. Here is our slide. Okay, and the specimen is over here. So what happened? So this is the light source. And here we have condenser. So condenser actually goes from here to all the way here. But from the down of the stage, you cannot see because condenser is actually covered with the Irish diaphragm. So when light comes out of the light source, condenser try to collect as much as light possible. So the function of condenser is to collect all the light that is coming from the source, and it actually gather all the light over here on the specimen. Okay, and what it does, it actually make a cone of light. So basically light is coming from the source. So many different light rays are going. So condenser basically collect all these light and it makes a light cone. Means all the light rays actually fall into this apex. This is the apex of the light cone, and all the light rays are collected over here. So the rays from the apex that actually illuminate the specimen, because the condenser and the specimen, they are very close. There is very little bit of gap, at the max half millimeter gap. So that specimen get illuminated from the light of the apex of the cone of the condenser. Now from there, light enters inside the objective lens. So from here, light enters inside the objective lens. From here, light all the way goes over here. So when the light moves through the specimen, it actually makes one image, maybe inside the objective lens, or maybe this area, somewhere inside the body of the microscope, somewhere. Okay, and that image is inverted image. Now, what happens when the light from that image goes through the ocular lens? Then ocular actually create another image inside your eyes. inside your cornea. Okay, so basically, and that image is much larger, okay, than the first image. So two images are produced by the microscope. First image falls either inside the objective or inside the body of the microscope. It depends on the structure of the microscope and the quality of the microscope. And the second image produced by the ocular, but it actually falls inside your eye, inside the retina. Okay, and that image is much larger than the first image. So that's the principle of microscope. If you see over here, you can see that's the light source. This is the condenser. You can see the condenser is really big and thick lens. So it collects all the light and it makes a cone of light. This is the apex that actually illuminate the specimen. From there, again, the specimen actually refract light based on its obesity. Okay, based on its opacity, not the obesity. Okay, so now objective try to collect as much light rays it can collect, and then it creates real image. This picture is little, it's wrong. Actually, it was in Word, and when I fixed it, it came down, and it should be actually here, somewhere here, from here to here, this area. Okay, that's the real image. Real image is much shorter than the virtual image, and that's the image. So now, from that real image, light enters inside the ocular lens, okay, and that increase, and that makes actually inverted image on your retina, and that's the virtual image. Virtual image actually falls into here. Okay, so this is the principle of light. We have so many different type of light microscope. Okay, we have bright field, we have dark field, phase contrast, interference, fluorescence, so many different types of microscope. Now, the bright field, at the first slide of this PowerPoint, I talked about the bright field microscopy. All these bright field, dark field microscope, all are actually type of light microscopy, where we use light to observe or to create the image. So all these are light microscopy, but how we utilize or how we use the properties of light, those are different from one to other, and that's how we have so many different type of microscope. So dark field, so bright field microscope, this is the characteristics features, this is the principle. Okay, so light cone is produced by the condenser. Almost all the light goes to the specimen, and almost all the light enters inside the objective lens. So basically, we are not losing any light over here. So that's the reason, if you remember the first slide, what happened? Both the specimen and the background is illuminated with the light. Okay, and because the specimen, it has different opacity at different area, different region of its body. So what happened? The specimen is little darker than the background. Background is really bright because we collected all the light over here. Okay. Now, in case of dark field microscopy, what we do, just before the condenser, we have a opaque light stop. So basically, in between, this is the condenser and this is the light source. In between the condenser and light source, we have a opaque stop over here. Opaque stop means that actually block the light. Okay, we don't allow all the light that is coming from the bulb We don't allow all the light to go inside the condenser. Okay, so that's the reason we have a block over here. We block that area, and that blockage is called opaque stop. Now, because we stop partially the light, so very little amount of light can go through the condenser. As a result, condenser make a hollow cone of light. Do you see? This cone is hollow. So this is hollow cone of light versus if you see over here this cone, you can see it's filled up with all the light rays. Okay, so that's the difference between dark field and bright field. So this part is hollow. We don't have any light rays. Okay, so very little amount of light falls onto the specimen, and from that specimen, all the light enters inside the objective lens. Okay, basically this part, all these sides, they remain dark. There is no light rays. Okay, so we only see the dark background because we blocked all the light. Light cannot enter inside the objective lens other than the specimen. Only light enters inside the objective lens is the light that comes through the specimen. So that's the reason for the dark field microscopy, the background is completely dark because we block the light. Okay, all the light that actually is supposed to go through the objective lens, there is no light. So background remains completely dark. We only allow very little bit of light that only can go through the specimen. So if specimen reflect, then that light can enter inside the objective lens, and that's the reason we can see you can see over here the specimens are really bright and the background is dark. Get it clear? So the difference between dark field and bright field. Bright field, we really do not have any stop, so continuous, and condenser collect a huge amount of light. So basically both the specimen and the background gets enough light to be illuminated. But for the dark field, we actually block certain amount of light. We only allow light to go through the specimen so that we can illuminate only the specimen. And that's the reason over here for the dark field microscopy, the background is really dark, but the specimens are really bright. I already talked about it. Phase contrast, almost like dark field microscopy. Okay, over here we have two area, two positions are there where we actually block the light. One just before the condenser, just like dark field microscopy, but in this case that we block this area with annular ring. Basically it is like a plate and it has ring, so it has hole actually, so light can move through this solid part. This area light can move through, cannot move through, but light can move through the hole. Okay, so that's the annular ring, just immediately above the light source. Another controlled area is just behind the or inside the objective. It's called faceplate. Over here also we block the light. We actually divert the pathway, light directions over here. So in this case what happened, again it's like from the light we block little bit of light, so condenser make a hollow cone of light. So not only only light that can goes through the specimen, they enter inside the objective lens. So up to this is similar like dark field. Now over here, so the light is actually coming from the specimen. But now here, what happens, we actually, some of the light, they can go continuously, spontaneously, they do not have any obstacle, but some portion of the light, they got distracted by the faceplate. So we actually change their direction. They move in different direction. So basically, we move the light in two different direction, two different pathway. So we actually change the phase of the light. Either they are in phase or some of the light are out of phase. So basically either they merge or they don't merge, they get separated. So two different types of light rays are there when ultimately we see through the ocular. Some portion they're really bright, some portion it's not completely dark, they have light, so it increases the contrast. Okay, so if you want to see the shape of the specimen, what we do, like the inside of the specimen, we make it, you know, really bright, and the outside that remains lighter, you know, area, lightly illuminated. So we can see that we increase the contrast to understand the shape of the bacteria. Okay, so that's one example how or why we use phase contrast microscope. So if the shape of the bacteria, it's little, we have a little bend over here. If this area is really bright, okay, highly illuminated, it would be very difficult to see that bend. But if this portion is lighter but this portion is darker, okay, that increase the contrast and you can see that bend area nicely. Get the concept? So phase contrast microscope, we actually do that. Electron microscopy, this is not light microscopy because we really do not use any light over here. Okay, so another thing I forgot to tell you, like light microscopy, you can see living organism. If you want to see dead organism, you can see. If you don't want to stain the organism, like if you want to see the natural color, you can see the natural color. If you want to give the color to increase the contrast, you can do the staining. So that's the light microscopy, okay? Bright field microscopy. For the dark field, you can see the living material because you cannot stain. For this dark field microscopy, we actually use unstained and live organism. And for the phase contrast also, live and unstained material we can observe. Electron microscopy, we don't use light. Instead of light, we use beam of electron. We don't have any lens. Instead of lens, we have mirror or prism. Okay, we have prism, so we can actually reflect the light. Or we have most of the time we have magnet. Okay, nowadays we really don't use prism, we mostly use the magnet. So magnet and electron. So electron replace the light and the lens replace the magnet. You really do not have to study in detail the principle. There are two basic types: transmission electron microscope, the TEM, and the scanning electron microscope, or SEM. Okay, TEM, it's if you want to see the tissue section and subcellular structure, so basically internal detail, then we use TEM. If you want to see the outside three-dimensional surface, then we use same. It's very easy to remember. I will give you a question like that. Okay, so if you want to see the internal detailed structure of mitochondria, which microscope would be the best? Light microscope, TEM, or SEM? Tell me. I want you to see the internal detailed structure of mitochondria.
And you're asking which microscope is better?
Yeah, and
Professor?
I gave you 3 options: light microscope, I gave you TEM, and SEM.
You wanted to see the
Would it be
we would
Tim?
use the, Tim.
We are going to use the TEM. That would be the best choice, right? Because TEM use the internal detail structure. Light microscope, mitochondria is so minute, we cannot really see in detail, right? And same is for the surface structure outside. So you can expect this type of question. If I give you that I want to see a ladybug movement, which microscope would be the best one?
Do you want to see the inside of the ladybug? Or
No, I want to see the movement of ladybug, the whole ladybug, and it is alive. I want to see its movement. If I want to see the movement, means it should be alive, right?
Okay, yes, ma'am. So then we would use them, right? Because you said it's
No,
surface.
no, no. Remember that TEM and SEM, these are the electron microscope. We are going to use the electron. That means we cannot use living organism. You can understand, electron means it will kill anyway, okay? So electron microscopy, we only can use for non-living material. So to observe the living organism, this is not the right choice. Okay, our best choice should be the light microscope. Because it's living, we really do not have to stain, and at the same time, we can see the movement of the organism. In this slide, there are it talked about different type of microscope, the principle of microscope. I actually went over almost all. I'm not going over it. Make sure to study all the different, but whatever the microscope you really need to study in detail, I talked about it. There are very few like fluorescence, confocal, just you really need to know in short. You don't have to go into detail. Okay, so the preparing specimen. What's the time? So let's keep today up to this. Okay, all the microscope and all. I am going to study from here, from the staining process and all those on Tuesday. Today is Monday. So Wednesday, I'm going to start from here. Okay.
Okay.
So make sure to read, if you can read the text tonight, that would be best, because you were here, I talked about it. It would be easier to get the concept if you read it maybe today or by positively by tomorrow, then you will remember and it would be easier to understand.
Yes.
So, do you have any questions?
No, professionally, I think I'm okay. Thank you so much.
Okay, you are most welcome. Okay, so we are done for today, and from this slide, I'm going to start it on Wednesday. Okay.
Okay, thank you, Professor. See you Wednesday.
Yeah.
Bye.
Bye.
Thank you. Have a good night.
Yeah, you too. Good night, everyone.
Good night, Professor.
Yeah, I will see you in labs and Wednesday this time. Okay.
Okay.
Thank
Bye.
you.
Thank you.